Welcome to NewYorkBIO's Emerging Companies Showcase, an event series partnered with the New York Stock Exchange to bring New York's financial community together with groundbreaking companies. In the first of two planned events for 2021, we are focusing on European and UK-based companies that could tap the US public markets over the next 12 to 24 months. We appreciate the sponsors of our event series, Woodruff Sawyer, Goodwin, Newmark, Moses & Singer, TriNet, and PHCBI. And now, we have a deeper dive with Martin Welschof, CEO of BioInvent.
So, Martin, it's wonderful to have you with us today, and we are really excited to have you presenting at NewYorkBIO's Emerging Company Showcase. This is an event that we've done with the New York Stock Exchange for the last couple of years, where we bring emerging, private or companies that aren't public in the U.S. Out to a broad audience of biotech investors, and we're really proud to celebrate exceptional science. This time we have brought companies out of Europe, and in recognition that innovation occurs everywhere, and it's something that we have an incredibly global industry, and we're genuinely excited that people are bringing new innovations to patients. So thank you so much for joining us. We're really excited that you're going to be presenting next week.
Yeah, thank you, Derek, and I'm very happy to be with you and tell a little bit about BioInvent.
That's excellent. So why don't we start with a little bit about your background? Why don't you tell us a little bit about where you came from and kind of how you got to BioInvent?
Yep, happy to do so. So I'm a scientist by training. I did my PhD and postdoc at the German Cancer Research Center. And since I was early on, I was more interested in applying science rather than doing basic research. So I joined the life science biotech industry, started to work for a biotech company in Germany. And from there, I moved then to the San Francisco Bay Area, where I spent some time. Worked there at Lynx Therapeutics, which became later Illumina. Probably you know that story. And from there, I moved then to Oslo, Norway, to join a private Norwegian biotech company, which I was running for roughly 10 years. And, it was a quite interesting story. We took it public through a reverse merger and then eventually sold it to a large Russian pharmaceutical company. And from there, they moved then to Dublin, Ireland, where I was running an Irish private biotech company for 7 years. And during that time, I was approached by BioInvent. They were looking for a new CEO that was probably at some time point during 2017. I think at the end of 2017, and then August 2018, I joined BioInvent. And the reason why I joined BioInvent were basically threefold. So first of all, I was very intrigued by the level of integration of the company, because BioInvent covers discovery of targets, antibodies, preclinical development in-house, own manufacturing process up to 1,000 liter GMP, and clinical development. So that means you really have a nice set of tools in order to move programs from the discovery quickly into the clinic. So that was one point. The second point is that Björn Frendeus, who was the CSO of BioInvent, he over the last probably a little bit more than 5 years, really developed an excellent preclinical portfolio, cutting edge science, well published. So it was a good starting point. And obviously I'm aware that, good science is not a guarantee for clinical success, but it's a good starting point. So if you start with crappy science, you will have for sure.
You'd rather start with it than without it.
Exactly. And then the third thing was that BioInvent is really, since the company is around for some time, a nice and very skilled team in antibody biology, from discovery through development, through manufacturing. So it's one of the leading experts in the world. And probably we come back to that later in more detail, also have a very interesting discovery engine. So the company started out as an antibody discovery company, became then an antibody and target discovery. It's a simultaneous process. And the way they run it is really that they screen on patient material directly, and then have a functional screening. So means they have a small molecule approach to the antibody library. Those were the things that attracted me to BioInvent, and I did not regret that I joined. So we achieved already great stuff during the last roughly 3 years.
Well, that's exciting, and it's certainly something that it's a story that I've seen personally, and certainly the ability to marry antibody discovery with target discovery and new biology is something that's great. And honestly, the level of kind of capabilities that you've described is rare in earlier stage companies. You usually get companies that are excellent at one of those things. You very rarely get companies that can do all three with a high level of confidence. And so, like you said, this company has been around about 20 years now, and that's it's a long history for a company to develop things like that. So can you give us a bit of a sense of where the company came from and kind of some of the iterations that allowed it to really develop those capabilities and get to where it is today?
Mm-hmm. So as I already indicated a little bit, so the company came really from the antibody discovery, and at the same time point they built a CMO business, manufacturing business. Mm-hmm. I mentioned the capabilities to produce up to 1,000 liter GMP. So the company can produce phase 1, 2, and 3 clinical grade material, which obviously is very good. And I think that was a starting point. And then the company basically was one of those companies having high quality antibody libraries, phage display. And I think they were probably one of the 5 best ones internationally, and still are. And that time they did a lot of antibody discovery deals, with Bayer, with Mitsubishi Tanabe, Daiichi, Takeda, just to mention a few. And quite interestingly, so all those companies that I've mentioned, they have now antibodies from the BioInvent and Coda library in their clinical development process, which is obviously nice because it gives you a nice background news flow without that you have to do anything for it because it's already in process.
Right.
And then I think the company also, to mention that, probably was too diverse because they tried already earlier to go into more clinical development later stage. And I think from my perspective, I think the mistake that was made at that time was it was too spread out. So it was HIV, it was oncology, it was cardiovascular. Yeah. And I think that was not generating really synergies. And then the company took a step back and basically started to develop their own portfolio, which is very focused in immuno-oncology around cells or targets that inhibit the immune system, basically. That's what the company is doing, and has built a very interesting, nice portfolio in that area. And when I joined August 2018, there was just one program in the clinic, which is our lead program that already has generated first very good data. And by the end of this year, we will have 5 clinical programs in the clinic. There you can also see what you can do with an integrated platform. Within less than 3 years to go from 1 to 5 programs is actually quite good.
That's very fast.
It's very fast, absolutely. And we will continue to be that fast because the engine is still running. And our current business model is really focused around to create those clinical development opportunities, show that they work, and then look into partnering. Basically, that's what we're currently doing.
Yeah. So it's really interesting because throughout the history of the company, and there's a reason this is a great story, it's, the company has hit on several of, the ways that biotech companies can become successful, but you kind of highlighted the reason that they're all really, really difficult. You start with a strong services business where you can build your own technology, and they did that. And then there's a strong discovery business, and those deals are actually really hard to get. So if you have a discovery business that you have to build up, if you have to do it in a short period of time, it's very, very hard. Like you said, it's great to have external news flow that you don't have to worry about, but it takes a long time to build that news flow. And then you said they had a portfolio that was spread out. Now, at the time, they probably said we have a diversified portfolio in a number of different disease areas, which is great on paper, but also carries its own challenges. And now you've gotten to the point where you kind of have these other things that have matured in their own ways, but you're now focused on a particular area that when you do something like that, you can do something like go from 1 to 5 programs in the clinic in the span of only 3 years. So it's an exciting story, and it's something where, many times it takes companies a long time to find the actual way that's going to make them valuable. And sometimes it depends on the market. Sometimes it just depends on external factors that people can't necessarily control. So what you have in front of you is really exciting because you're working in immuno-oncology. And it's an area that, I think we can probably call, revolutionary, certainly over the last decade. We've seen, advances in and in cancers like melanoma that were unheard of beforehand. So it's excellent. But there's also the challenge of patients that don't respond, right? We don't know, it's not a large fraction of patients that do respond to immunotherapy. And we don't really have a great handle on who's going to respond beforehand. So can you talk a little bit about what differentiates BioInvent's programs and how you really are going to try to help those numbers?
Yes, absolutely. So I would start maybe explaining a little bit the platform, not into much detail, but I think it's a very important part of the journey for those various programs. And what Björn Frendeus and his team has done, they have built a very tight collaboration with the local hospital in Lund, where the company is based, such that we receive a few times per week fresh patient material. And that patient material obviously doesn't have to travel far. So it's basically a bike ride of 5 minutes, and then it's back in the lab. And then we start the screening. And we really start or do the screening on those cells directly, or tissue directly, without any further modification. So that is the only thing what we do is QA/QC to ensure that the material is alive and kicking. And then we take it into screening. And that, of course, is a very important first step, because you do not use that, let's say, literature and say, well, what could be an interesting target? Then you put it as a peptide, or if you just screen for a specific epitope or recombinant protein. No, you really screen on the culprit cells, basically.
Yep.
And then the first step is then, obviously you need a high quality human antibody library, which we have. It's the encoder library. And then out of that library, then we select specific binders to those cells of interest. And then we clone those binders directly into full length IgG. And before we check for the target, those IgGs go directly into functional screens. So they go through a number of very challenging animal models to check them or characterize them regarding their therapeutic properties. And then we would only focus on those who have shown strong therapeutic properties in those models. And for those, we also identify the target.
Right.
So the reason why I mentioned this, so that means all the antibodies that we have generated for ourselves, as well as for collaboration partners, such as most recently for Pfizer, are preclinically validated regarding the target and also regarding the binders.
Yep.
And the approach is very powerful. So normally you would get a panel of antibodies per target. So would get antibodies with binding to different epitopes that would translate into different mode of actions. We get blockers of ligands, inhibitory antibodies, agonistic antibodies. So we get the full panel, such that we can really do a nice selection for those that we then take into clinical development. And then, of course, it's very important what you screen on. So we had a very strong focus on T regulatory cells of the tumor microenvironment, because it's a very important cell class. Another focus that we had is on tumor-associated myeloid cells, which is another very important cell type. And in cases, so in the case of T regulatory cells, we focus on antibodies that are very efficient in depleting T regulatory cells, specifically in the tumor microenvironment. And I can give you later examples. On the tumor-associated myeloid cells, we try to discover two different categories. First of all, we discover antibodies and targets that allow for depletion of inhibitory tumor-associated myeloid cells, but we also have found targets and antibodies that we can use to re-educate, basically turn an inhibitory TAM into an activating tumor-associated myeloid cell.
Right.
So those are the two cell types that we're currently focusing on. And I guess we come later to the portfolio. And then we have one program where we specifically focus on a very interesting target, which is Fc gamma R2b, which is the only inhibitory receptor on cells of the innate immune system. And that has generated two different antibodies and will be at the end of the year 3 clinical programs.
Yeah, that's exciting. So you're basically taking the elements of biology that the tumor uses really as its own defenses to either evade the immune system or to cloak the immune system. And you're kind of turning those things, hopefully, to attack the tumor itself.
Correct.
Yeah, that's really exciting. That's exciting. And I think one of the things that we've seen over the last probably 5 to 10 years is a better understanding of the roles of individual cell types, either within the tumor microenvironment or even on the outsides of the tumor, that affect how the body sees it, how the immune system sees it. and how you can mount an offense. I think the really interesting thing that you're talking about here is that your targets and your discovery start with patients. So you don't have to, you will eventually have to translate it back from animal models into a patient model, but you've really started with something that is a set of biological targets that are expressed in their natural state. I'm interested to hear how you tackle the need for patient diversity, because very oftentimes these samples are difficult to get. So building up an actual set of patients that kind of spans the diversity of either tumor types or different modalities within a single indication is really challenging.
Yeah, but I think, and one thing before I answer your question directly, what I forget to mention, so obviously what we screen or get out of this process is human antibodies that can be used as therapeutics.
Yep.
What we do in parallel also is run a so-called mouse track. That means we also generate the mouse derivatives such that we can check, because we very often have immune modulatory antibodies that we would like to check in the mouse system. So we need to have the mouse counterpart. So we have both.
Right, you need to run it in parallel.
And mouse track, so we do that in parallel. Regarding the materials, actually that's a very important and critical part, because the collaboration that Björn and his team were able to set up is very broad. So we don't suffer that we don't get enough material. So we can, when we run a campaign, we can pool quite a number of patients, such that we make sure that we cover diversity, and also make sure that we identify markers. And later when we talk about the programs, you will see that we have that makes sense for a broad patient population rather than to be very, very specific. So we could be very, very specific, but so far our screening campaigns that we were running are broad screening campaigns to make sure that we have targets that are really interesting commercially, since they are interesting for a high number of or broad range of patients.
Yeah. So you've mentioned the collaboration with the hospital a couple of times, and I think this is a good segue here. So can we talk a little bit about really the environment in Sweden for starting companies, for bioscience companies in general, and for collaborations, because those are really the things that can help take research from the lab into something that can then be clinically commercialized. And in the US, we hear all of the stories between San Francisco, San Diego, Boston, and more often than not, now we're starting to hear more and more out of New York. But I think there's an appreciation for the fact that innovation can come from everywhere, and it's really good to get a sense of how it works in other places.
Absolutely. And so as you maybe have gathered from my CV that I briefly was summarizing at the beginning, so I've spent quite some time in the Nordics. So started in Norway, where I'm also still living as of today, worked some time in Denmark, in Copenhagen, and then now since almost 3 years in Lund, Sweden, and have seen how this whole environment has developed. So I think Sweden and Denmark were always quite leading, also on a European standard, because they had, two very, very good and successful pharma companies. In Denmark, obviously Novo Nordisk, and in Sweden, Astra, which then became AstraZeneca, which I think was very important. Both countries had a lot of support for academic research in any way. There's the Karolinska Institute in Stockholm, for instance, which is world renowned, and then you have also a couple of very good institutions and universities in Denmark. Norway was lagging a little bit behind, since it was very occupied, at least during the early strong economic years, by gas and oil, obviously, because Norway is a gas and oil producing country. But I would say has caught up very nicely during the last at least 10 to 15 years, so that you really can talk about Nordics. Obviously, you have the area, Copenhagen, Malmö, Stockholm, which is very, very strong. And then you have also Oslo, where you have the Oslo Cancer Cluster, and they're all collaborating, those different regions. And I think there's one important characteristic. It's like in the US as well. So there's strong support for excellent academic research, And I think also now, a lot of support to translate that into companies. So, and there are a couple of success stories. If you think about Norway, there was, for instance, Algeta, that was one of the big success stories that was purchased by Bayer. And of course, then many companies in Sweden as well as in Denmark. So I think, and people are not shy. People mean coming from academia to really translate their science into something more commercial. And this has now a strong and long history of at least, I would say, 25, 30 years. Which has led to really a very, very successful cluster. You mentioned the clusters in the US, and I think we have a Nordic cluster in Europe. Yeah, so which is Oslo, Lund, Stockholm, Malmö, and Copenhagen, to mention the most important ones.
I think the other thing that's interesting about the Nordic countries is there's a pretty good level of pipeline and support from the public markets. So there's a good path for companies to get started and be able to tap public market capital and actually be able to build companies, and perhaps a slightly different way financially than they're built in the US and in perhaps other countries. But at the end of the day, capital is capital, and however you get it into the company, the real main thing is that you're able to advance your programs and deliver medicines to patients.
Correct.
Excellent. So switching back to BioInvent here, I'd be interested to just hear from you about where you see the company going over the next 5 years. You've got an exciting pipeline, you've got things in the clinic, and where do you see the company going, and how do you see it transitioning from hopefully a clinical stage company into something that's commercial and delivering medicines to patients?
Yeah, so we have currently 4 programs in the clinic, and by the end of the year, there will be 5 programs, targeting those mechanisms that I mentioned briefly at the beginning. The first program already has shown, an early proof of concept, and that's a program that we also have partnered geography-wise already for China with CASI Pharmaceuticals, probably well known, NASDAQ listed in the US, but with focus on commercialization in China. We also have a clinical supply and collaboration agreement around that program with Merck. And then we have a couple of other programs which are all roughly in the same stage, phase 1, 2. So the first one, as I said, has shown early proof of concept, and the others are close. So basically, over the next couple of years, we will have a number of interesting clinical development milestones. And the way we structure our programs is as such that we, on one side, tap into really large unmet medical needs, but also have as part of our clinical development also niche indications, small indications, where we can go for conditional approval and have a fast track to market approval, which is of course interesting. This we have for our lead program and all for our program called BI-1808, which is targeting TNF receptor 2, which is one of the hot and upcoming targets in immuno-oncology. All the programs are first in class, which I think is very, very important. And that's also why we have a portfolio. So we're just focusing one program, and we'll continue to focus on the portfolio rather than one specific program, at least for some time, and basically get further support in the clinical development with good partnerships, which would generate further income, but also split the risk or diversify the risk a little bit further, early validation of the programs, and getting help and support regarding commercialization of the programs. The engine that I described at the beginning is still running. So, those 5 programs that we have by the end of the year in the clinic, we are already supporting them from below. So there's currently no end really. So we can, I would not say unlimited numbers of targets and programs. There is a limit, of course, but not yet. So we can dig deeper and find new interesting assets. The other thing, our current focus is clearly immuno-oncology. The platform would have also great applications in autoimmune and inflammation. And we are tapping a little bit into that as well and see what we can do there. Saying so, of course, the key message to our shareholders, we remain focused, so it's immuno-oncology. But if you can see that we can do there something as well, then then we should also build a strategy around that in order to really get the interesting targets and antibodies that would help for those disease areas as well, since we have this nice functional screening approach.
Yeah. Well, listen, I think that's a great story. I mean, if we look at what ASCO has kind of shown us this year, you had kind of the readout of the checkpoint inhibitors against things like LAG3, TIGIT, and I think TIM3, and those targets have been around for a while. And while there was good data, I think one of the take-home messages is we haven't cracked it yet. We haven't cracked tumor biology. So I think the fact that you have a number of things that are first in class, and you're actually starting with function off of human tissues, I think that's a really great base to start from. I also love the fact that you brought up inflammatory and autoimmune conditions. I think one of the hypotheses that have been around for a while, that is, if you take kind of the checkpoint antagonists that we're using in oncology, and you flip the switch the other way, you turn them into agonists, you actually have real opportunities in the autoimmune space. So I think you're doing something really, really exciting. We're excited to have you be part of our event. I can't wait for you to present next Wednesday. And I wanted to thank you for being here with me today, and wish you good luck.
Thank you very much, Derek.
Thank you for joining us on A Deeper Dive. We're looking forward to the Emerging Company Showcase on June 23rd. Special thanks again to our sponsors, and for more info, visit us on www.newyorkbio.org or on Twitter @NewYorkBio.